Thanks for the kind invitation and apologies for the technical mishap there. So I'm going to start this by talking about one of the ways that we've been using resting state fMRI to characterize subtypes of depression and how this might be used to identify the who question or figure out the who question. Who might be the best person for TMS, in this case for treatment-resistant depression? I'll then also show some unpublished extensions of that in individuals treated with SSRIs. And then we'll close out this talk just by talking about maybe pitfalls or problems if you wanted to try and use this approach with your own data. And I don't have any disclosures. So to start, depression is an extremely heterogeneous disorder. You need 5 of 9 possible symptoms from the DSM-5 to be diagnosed with depression, which yields numerous combinations of symptoms under the same diagnostic umbrella. And as a result, it comes as no surprise that two different people will, with different symptoms, won't necessarily respond to the same intervention. And so many decades have been spent thinking about ways of tackling this symptom heterogeneity. Originally, it was from this top-down based approach on the right, where we take 2 people with a similar subset of self-reported symptoms, lump them into specifiers as outlined by the DSM, and see if there are any neurobiological differences between these 2 groups. And as a result, that hasn't really yielded prospectively meaningful assignments in terms of treatment outcomes. What us and others have turned to is a more brain-based bottom-up approach, which you can see on the left, where we try to identify subgroups, homogeneous subgroups based on either patterns of brain dysfunction, genomics have been thought of as well, and then using those subtypes to better understand who might be best suited for one treatment over another. And it's not just me that's been doing this. Lots of others are also quite keen to understand subtyping, not just for TMS outcomes, but for also things like resilience following trauma, psychopathology more generally, or mood disorders or anxiety disorders more broadly. And there are a number of different methods that we can use to try and understand this heterogeneity. In our case, we've been using something called canonical correlations analysis, or CCA. This is a multivariate approach in which we can take baseline neuroimaging features. In this case, we use resting state functional connectivity. We can take symptoms as reported by a self-reported or assessor-rated scale. And what CCA will do is try to find linear combinations of these 2 modalities that will maximize their correlation. And that correlation between these 2 scores, the resting state score and the clinical score, is called the canonical correlation. We can project new data into this CCA space using the coefficients to derive these linear combinations and assess new data's performance using that canonical correlation. So the stronger the canonical correlation, the better our latent dimension is at explaining co-occurring symptoms and patterns of functional connectivity. And about 8 years ago, we tried this in a large sample of individuals with depression. We were able to find 2 dimensions of co-occurring symptoms and functional connectivity. And using hierarchical clustering, we were able to delineate 4 subtypes of depression. These were stable if someone was still depressed a month later and you scanned them again. And subtype assignment did predict response to TMS. In this case, it was dorsomedial TMS. Now, there was a subsequent replication attempt by an independent group, and unfortunately, at the time, that independent replication was not able to delineate the same 2 dimensions or subtypes using hierarchical clustering. And they raised some pretty important limitations of our original approach, which I will highlight. The first is sample size. And so, we used a multi-site data set at the time, which may have limited our ability to generate stable or generalizable subtypes or dimensions of symptoms. Another issue was overfitting. Canonical correlations analysis is really good at finding patterns in your data, and we didn't use regularization at the time, so applying a regularization scheme is probably one that would improve generalizability. And then lastly, we used only 16 items from a depression scale called the Hamilton Depression Rating Scale, which was designed in the 60s. It's kind of a gold standard for assessing outcomes to treatment. However, it kind of conflates some symptoms together, or it kind of doesn't include other symptoms like sleeping too much or eating too much. And one example of this, which I like to highlight, is often anhedonia, which is deficits in reward processing. There's one item in the Hamilton that looks at this, the work and activities item, but it, conflates multiple aspects of hedonic function like liking and wanting, as well as productivity at work or, other aspects of day-to-day function. And so we're somewhat limited with what we can do with just those 16 items of this scale. So we recently published an update in response to some of the important limitations that arose from the discourse of our initial paper. And so we sought to first assess the performance and stability of regularized CCA and clustering, hierarchical clustering in depression. We sought to further characterize these dimensions and subtypes, and we also wanted to see whether or not our updated model would also predict response or remission to TMS. And in this case, now we also have people not just treated with dorsomedial TMS, but also dorsolateral TMS, both 10 Hertz stimulation once daily and theta burst, intermittent theta burst once daily. And this sample of 328 individuals were all scanned on the same scanner using the same neuroimaging parameters and in the same way, which was sort of overcoming the hurdle of a multi-site sample. To classify our dimensions, we still used the 16 Hamilton items, but we also added a few additional items delineating things like liking and wanting, and included other items such as irritability to sort of better characterize some of these dimensions related to depression heterogeneity. Now, because we used regularization, we needed to first optimize how much to regularize our model. And in this case, we used a nested cross-validation scheme where we first subdivided our data into a 2/3 training and a 1/3 test set. And we left that 1/3 test set alone until the very, very end. That 2/3 training set was further subdivided into a training and a test set. And with that inner training set, we attempted a grid search across 3 different features to optimize, or parameters to optimize. The number of features included in our model from the resting state side of things, as well as regularization of our 2 modalities. And that was done in a grid search way, training and testing with that inner loop only. Once we figured out what was performing the best in that inner training loop, as quantified using the canonical correlation that I described earlier, we then combined that inner training and test set again and tested the optimal combination of hyperparameters on that left out 1/3 of the data set. We did this 100 times. And so what did we find was in that inner training and test set, the optimal hyperparameter combination was selected 98 out of 100 times, and our outer training canonical correlation in that outer test set was almost 0.5, which is pretty high. The training canonical correlation was about 0.8 in the first canonical variant, or the first latent dimension. So that's only assessing the very first latent dimension of CCA. We can also see whether or not we can perform well in subsequent latent dimensions. So we assess this in 2 different ways. We first, again, split the data into training and testing, trying to figure out how many canonical variates we could reliably get a robust canonical correlation in. And we found that the first 3 canonical variates or dimensions were performing significantly well relative to permuted data in unseen sample data. We also tested the stability of our model. So this was how the different latent dimensions were correlated with specific symptoms. And again, the first three canonical dimensions, or latent dimensions, were significantly stable, meaning that from subsample to subsample, the same clinical characteristics were being pulled out from one in each latent dimension. So what did these three dimensions look like in the full sample? Well, the first canonical variant had significant clinical loadings on a number of different clinical symptoms, such as increased mood, somatic symptoms, a little bit of anhedonia, and some anxiety, which was associated strongly with basal ganglia and thalamic connectivity. A second canonical variant was related most specifically with anhedonia. And it was associated, rather surprisingly, to higher order visual regions, although we did see some other regions such as orbitofrontal cortex, dorsal ACC, and dorsolateral prefrontal cortex associated with this dimension as well. We also found a third dimension related to insomnia, which loaded strongly on sensory motor regions implicated in sleep. Interestingly, a reviewer asked us to look at medication use, and we actually found that this dimension was related to sleep aid use. So if you're using like zolpidem or zolpiclone, which are sleep aids, Z-class drugs, you had significantly different loadings onto this dimension, which will make sense in a second. These dimensions had fairly distinct patterns of functional connectivity associated with them, and they weren't necessarily abnormal resting state fMRI features. So not all features that explain heterogeneity in symptoms are abnormal. So we then sought to test the stability and performance of a clustering solution using hierarchical clustering. In this case, what we found using the Kolinsky-Haribas criterion was that a 4-cluster solution was stable, or was the best performing clustering solution when we tested 2 to 10 cluster solutions. We tested that against permuted data, and it was better than what we would expect by chance. And our clustering solution was significantly stable, indicating that from subsample to subsample, the same people were being clustered together. And this is just the dendrogram showing that 4-cluster solution. Now, I mentioned that third canonical dimension was associated with sleep use, and when we tried clustering people with all three dimensions, it did not perform very well. And as a result, we're actually using only the first 2 dimensions here. So we weren't totally sure why that was the case before the reviewer was like, hey, take a look at medications. So it kind of was a nice explanation as to why the 2-dimension solution worked so well during clustering and the 3-dimension solution did not. Now, these subtypes differed quite considerably in terms of symptom severity. I'll just walk you through some of the trends. Subtypes 1 and 2 had the most severe symptoms related to mood and somatic symptoms like fatigue or decreased appetite. Subtypes 2 and 3 had the highest self-reported anhedonia and irritability. There were some differences with respect to anxiety between those 2 groups, and subtype 4 in red was the least severe overall. These 4 subtypes also had different patterns of atypical resting state functional connectivity relative to controls, with the biggest differences by subgroup related to striatum and thalamus, orbitofrontal cortex, and dorsolateral prefrontal cortex bilaterally. And then in terms of TMS response. So as I mentioned, the sample were all people referred for TMS. About half of them received dorsolateral, left dorsolateral, another half received dorsomedial, and again, a variety of different kinds of protocols, 10 Hertz or theta burst. And similar to our original findings, we did see that the subtypes with higher rates of anhedonia or deficits in reward processing had lower rates of response or remission to TMS. And this was seen not just with our categorical subtypes, but also continuously with that anhedonia dimension scores. Interestingly, this finding was consistent across the board when we looked for differences by stimulation site or even with protocol, TMS, or 10 hertz versus theta burst. So this was pretty stable across the board for all the different kinds of interventions that we delivered. So this was all just with the data that we published last year and with TMS. What about new data? Can this actually predict response to other interventions with unseen data? And we were really fortunate that we had access to people treated with SSRIs with resting state fMRI. And all we did was we tried to project this data, or the SSRI data, into this model and assign their subtypes. So they were never used for training. They're actually different scanners, different imaging protocol, but we tried our best to standardize them as best we could relative to the parameters from our original data set and just simply assign subtype assignments to these folks based on their projected canonical variate scores, and see just whether or not there were differences by subtype with respect to response or remission to SSRIs. And we actually found that. So we first found that aspects of the clinical loadings that we saw in the dimensions were replicated for both the first two canonical variates, such as mood and suicidality associated with canonical variate 1, and anhedonia and aspects of anxiety in canonical variate 2. And subtype assignment did differ with respect to remission to an SSRI. In this case, it was 8 weeks of escitalopram. So just to walk you through this, subtype 1 fared really well to both TMS and SSRIs. Subtype 2 had the highest response rates to SSRIs. Subtype 3 did poorly overall to both treatments, and subtype 4 did quite well to TMS only. There was a recent meta-analysis published looking at adjunct SSRIs with TMS, showing that relative to SNRIs, another antidepressant class, SSRIs in combination with dorsolateral prefrontal cortex may yield some adjunct benefit. So what this might give us is a really kind of exciting testable hypothesis that individuals subtype to group 1 might fare quite well to adjunct SSRIs with TMS. So I'll wrap up this section by just saying that subtyping approaches can be both stable and robust given the right data set. We found that not only was it stable in holdout data within our training set, but also to new data in individuals treated with SSRIs. And though I didn't show it here, it is also stable longitudinally. The TMS sample did have post-treatment scans, and we were able to show that subtype assignments were stable even in those with remitted depression after TMS. Now, what if you have a large, like a data set that you want to try this on? So I want to highlight some of the ways this can go wrong, because this is a pretty powerful approach when you have the right data set. So I kind of want to make this part interactive, if we've got the time for it. What do you think might cause this to not work, this kind of approach? Anybody have any ideas?
Small sample size.
Pardon?
Small sample size.
Yeah, small sample size. So yes, definitely sample size is one of them. So we did test that in our original training set of 328 individuals, where we just took we just took how small we could go to see whether or not our model was still stable or producing generalizable results in test data. And as you can see here, the first three columns are just the regularization parameters that were optimal given the sample size that we had. So we did the same nested cross-validation that I showed before. This is the proportion of training iterations that selected this optimal combination of hyperparameters. And these tell you what the performance was in test data in the outer loop. And as you can see, at n of 50, it's never stable. And as you get a larger and larger sample size, we do see incremental improvements in stability as well as in performance. With an n of around 250 in your full sample, giving you a stability of 0.7 and pretty comparable performance in the first canonical variate in outer test data, which is 0.37. Anybody else have any ideas? I mentioned a few over here, inclusion and exclusion criteria. So if you're using multiple data sets from different, say, consortia, There could be differences in inclusion or exclusion criteria that could impact the quality of your results. If you're going to go with a multi-site data set, how are you going to handle the batch correction? Was the imaging harmonized in any way during acquisition, or are you going to statistically harmonize the data? So are you going to use the data set itself to harmonize it, or do you have a sample of healthy controls that you can leverage to model side effects, similar to what folks at, I believe, UPenn are doing with crystal Zikos? Any other thoughts about how this could go wrong, other than sample size? Yeah?
Like lack of clear boundaries between subjects and the canonical space.
Yeah, so if you're projecting new data, finding, or if you have limited, say, range of, is clinical severity, is that what kind of you're going for?
Yeah, or
Maybe
if it's
clarify?
just like a Gaussian distribution and there's not a clear like kind of centers for means or hierarchical.
Yeah, absolutely. And I mean, certainly, like, our clustering solution was significantly performing well and stable, but if you actually look at the CCA space, and I think I showed that previously, like, the boundaries are not very clear. Like, yes, it's significant, but probably the continuous measurements are more useful, in my opinion. However, clinicians really like categories to make decisions, right? So having both and showing that both work is kind of an important thing for sure. Anything else? What might not make this not work very well in your data?
Population skills and the listing MRI can change the result.
Absolutely. So we've only, in this, what I've shown is all the same power ROIs that we originally used in the Drysdale paper, but definitely performance. Pre-processing pipelines could make a big difference, and the selection of parcellation is something that might be worth rigorously testing systematically. Definitely. Anything else? Anybody else have any ideas? Okay, I will provide you with some more. Okay, so another big problem is our CCA, or canonical correlations analysis, requires complete data. So you can't have any NANDs in your data. So how are you going to figure that out? Are you going to remove participants from your analysis? Are you going to impute? How are you going to do that with by avoiding data leakage? So you don't want to cheat in any way or peek at your test data. So thinking about that is something that's really, really important. What if you have like different data sets from different folks and they have different scales? I mentioned the Hamilton Rating Scale for depression. I think somebody else, Maria, mentioned the MADRS. Both are very well used and loved depression scales, but how are we going to combine them? And that's not trivial either. In my lab, what we're trying to do is use AI and item response theory to try and figure out which items from scales are similar to each other. So using AI and sentence pair regression, using encoders essentially, is one way that we can figure out what items are kind of similar to each other, and then use those quantitatively using something called item response theory. And then also thinking about what other kinds of modalities or aspects of cognition or behavior are at your disposal. So Leanne Williams over at Stanford uses behavior instead of symptoms to look at subtypes of depression. We're also using the Hamilton, which is the average symptoms across a 7-day span. What if you have longitudinal data? Behavior, actigraphy. The possibilities are endless. One way we've been thinking about this is not just using baseline severity, but also change in symptoms to a treatment. So I'll give you an example. This is the MADRS for 2 people who received SSRIs. The MADRS has 10 items. Both of these people had the same MADRS score pre-treatment and post-treatment. And they also had a 50% improvement to escitalopram, but you can see that the item-specific change is very different. And so we've been using CCA to try to delineate subtypes of response. Items, symptom-specific improvement, and co-occurring sort of baseline functional connectivity predictors. We've been able to find 3 subtypes of SSRI response that way, which had differences in like anxiety and mood and neurovegetative improvement, as well as response and remission. And we were able to independently replicate this in other data that was not included during training. Another problem sometimes when we're using these larger consortia data is, again, this is related to exclusion and inclusion criteria, but a big problem is limited item range. So sometimes these cross-sectional studies will use like samples that might not be too severe, or biomarker studies will exclude the most severe due to safety concerns. And this is especially a problem when we're thinking about suicidality. Often items like a Hamilton are used to exclude individuals, or it's clinician assessment, to exclude people with active suicidal ideation or intent, which means that our biomarkers are not going to be, or subtyping assignments are not going to be very robust for these kinds of symptoms. And so these are 3 samples that I've had the good fortune of using in the past. And this is just severity on a single item that assesses suicide, suicidal ideation. And as you can see, it's very skewed towards 0. And if you actually look at it, only about 17% of the sample are actually suicidal, and only 2% of them had actual intent. So we're really skewed against people with passive ideation who are thinking, maybe I don't want to live, or I would be better off dead, or they have no ideation at all. The subtyping, the data set that we used for training actually had a much more uniform distribution across this item, for example, and as a result, we did see nice stratification of suicidal ideation by subtype. A couple more slides. Another thing is univariate correlations. So one thing that we like to do before we even start an RCCA model is to see whether or not the just simple correlations between our symptom data and our resting state are actually robust enough to detect multivariate associations. It's kind of a garbage in, garbage out situation. Not saying that anybody's data is garbage, but if you're not seeing that those univariate correlations are stronger than what you would expect by chance, you're not going to get very stable results. And as a result, we did check this. So this is in the supplement of our paper, where we did just take univariate correlations, or we did feature selection, removing the best performing features, and just had a look at whether or not model optimization was stable or not. And we did see reductions in stability and performance as a result of taking weaker univariate correlations. Last couple of slides. The one thing I'll mention too is data leakage. So we use this nested cross-validation scheme to avoid peaking and overestimating the performance of our model. So that's one way during optimization and training that you can do to try and get a precise estimate of how well your model is performing. And then the last thing is obviously independent validation. So if you do have extra data lying around or collaborators willing to share, that's another way to really get at the robustness of your findings. And then the last is just different modalities. So I showed everything with resting state fMRI. Our lab is also interested in looking at this in the context of brain aging, and also integrating some of these findings with EEG. So stay tuned. We're just starting these projects now. So I'll stop there. Thank you all for your attention. I'm happy to take questions. And if you're interested in learning more about the subtyping assignments and everything, I'll be giving a talk tomorrow.